All right. Good afternoon. My name is Shreya, and today I will be discussing the impacts of microbial exposure on infant immune development. So to start with some background information. Despite significant advances for infant health in recent years, infectious disease still remains a leading cause of infant deaths worldwide. This is partly because during gestation, the fetus develops in the relatively sterile environment of the mother's womb, so immune tolerance between the mother and the fetus is of greater importance than immunity to pathogens. However, at birth, the newborn is suddenly exposed to pathogens, and it takes many months for the immune system to fully develop, leaving many infants vulnerable to infection. To resolve this issue, effective vaccines are necessary. However, many vaccines are less efficacious in infants because they're designed for a mature immune system in mind. And this is largely due to limitations of current animal models, which simply makes it difficult to research many of the mechanisms of protective immunity within infants. And one reason for this is many current mouse models neglect microbial exposure. And there is compelling evidence that suggests that microbial experience can actually change the immune response of laboratory mice. So, in an attempt to make a more accurate model, many researchers have begun developing so-called dirty mouse models. So most current research utilizes mice housed in specific pathogen-free conditions, as you can see here. These mice are called SPF mice and are often used to represent a benchmark of studies of the immune system. So as a result, these SPF mice are exposed to very few pathogens to ensure that specified diseases don't interfere with the experiments, primarily for reproducibility. However, because of this limited pathogen exposure, these current models exclude the role of microbial experience in the development of the immune system. Now, clearly, most humans are not actively excluding microbial exposure in their daily lives. So this has raised some questions about the accuracy of using an SPF model for humans. So to combat this issue, researchers have developed various methods to introduce pathogen exposure to mice. For example, sequential infection. Another method is co-housing, which involves a pet store mouse or a mouse with pathogen exposure. And this visual from Embracing Microbial Exposure in Mouse Research by Huggins et al. Provides a very good explanation of this co-housing process. Essentially, the pet store mouse is co-housed with the laboratory mice. And numerous studies have shown that co-housing these mice for 3 to 6 weeks can actually change the immune response of these adult SPF mice. And these mice with normal microbial exposure, they're shaded in these red lines, are called enemy mice, or normal microbial exposure mice. So I was interested in combining the information surrounding the infant immune system with the influences of microbial exposure. And this led to the development of my research question. How does microbial exposure impact infant immune maturation? So in terms of surrounding literature, I read numerous papers discussing the introduction of microbial exposure to laboratory mice. However, these two papers stood out to me the most. The first paper utilized the co-housing method, and they compared memory CD8 T cell differentiation between mice and humans. So in this, Guerra et al. Characterized the CD8 T cell distribution in normal cervical tissue in humans. And they noticed that in adult SPF mice, CD8 T cells were almost completely absent from those same cervical sections within the mice. However, in pet store mice, pet store mice exhibited CD8 T cell levels similar to that of humans. However, these pet store mice had a different genetic composition than the laboratory mice, so they utilized co-housing to influence or transfer pathogens, and noticed that these co-housed mice also exhibited similar CD8 T cell levels to that of the humans. So from this, Guerra et al. Determined that the pet store mice and co-housed mice recapitulate aspects of human CD8 T cell differentiation that are often absent in laboratory mice. The second paper took these findings a step further by investigating SPF and enemy mice response to infection. They infected the mice with Listeria monocytogenes, and Huggins et al. Observed the immune response. They noticed that the enemy mice controlled the infection at a faster rate than the SPF mice, indicating that a more developed and mature immune response was there in the enemy mice because of their microbial exposure. So my literature review showed me that microbial exposure leads to a more developed immune response in adult mice. And it also provided me with the layout of a study like this, as I gained a better understanding of the co-housing method, since both studies use co-housing to analyze different aspects of microbial exposure. So now that I've discussed some of the backgrounds of the project, we can move on to the methods. So to understand the impact of microbial exposure on infants' immune development, I employed a quantitative research method by analyzing collected data. And to do this, I worked with Dr. Schultz at the University of Minnesota. Unfortunately, due to the pandemic, I was unable to enter the lab. However, Dr. Schultz walked me through the data collection process. So to give a brief overview, breeder pairs of laboratory mice were housed in the SPF facility. And then other breeder pairs were housed in the dirty mouse facility with a pet store mouse, shaded in red, to generate enemy mice. And these mice were sacrificed at 2 or 3 weeks of age and run under flow cytometry to gather their immune cell compositions. So generally the co-housing method has the mice co-housed for 3 to 6 weeks, and all the papers I read about co-housing used it in adult mice. However, it had to be modified for these infant mice because, if we're using 2-week-old mice, it's impossible to co-house them for 3 to 6 weeks. So instead, the adult mice, or the breeder pairs, were co-housed with the pet store mouse, so that the pups that were developed from these enemy mice the parents gained microbial exposure, and the pups gained maternal immunity, as well as the microbial exposure from the pet store mouse. Preliminary data was also collected in the lymph nodes and spleen, and it revealed significant differences in the frequency of macrophages between the SPF and enemy mice. As a result, a more intensive panel was introduced to include inflammatory monocytes. Also, the preliminary data collection only looked at frequencies for certain cell types, so we then decided to include the actual number of cells, or the counts, in the next data collection, because it could provide more information about the cell compositions. And as you'll see in the results section, interestingly, the frequencies and counts of these cells often had very differing trends. So the initial immune cell compositions were quite overwhelming, as they displayed the counts and frequencies of over 32 different cell types. Therefore, I decided to focus primarily on the memory cell compartment and inflammatory monocytes. Many of the previous studies I read discussed the memory T cells, as well as inflammation, often referring to like the inflammatory cytokines. And for example, like Buera et al., they included the CD8 memory T cells. So that's why I decided to focus on these two sections more specifically. I'd already developed the background knowledge, especially with these cell types in the adult mice, and so I thought that'd be best to carry that over into these infant mice. So after obtaining the immune compositions, I analyzed the data using GraphPad Prism 9. I initially began with a two-way ANOVA because I was comparing both different treatments and different ages. However, I quickly noticed that the two-way ANOVA only gave information about the difference between the treatments and the difference between the ages. So I couldn't compare both treatment and age at the same time. Like if I wanted to look at difference in treatment, it combined both ages and only looked at the SPF versus enemy. So it wasn't able to illustrate the differences at individual time points. So I decided to use a student t-test instead. I compared the SPF and enemy data individually for 2 and 3 week age mice using the unpaired parametric t-test, and p-values of less than 0.05 were considered statistically significant. So now some of the results that were found. I'll sort of go over the most important results, as these graphs tend to speak for themselves. But as you can see, 1 star represents a p-value of less than 0.05, 2 stars would be a p-value of less than 0.01, and then 3 stars is less than 0.001. So the graphs are sort of organized such that the counts and frequencies of the central memory T cells are the first two, and then the counts and frequencies of the effector memory T cells are the last two. So I generally noticed less significant differences within the spleen than in the lymph nodes. So just to begin with these CD4 memory T cells in the spleen, the number increased similarly between the SPF and enemy mice. However, the frequency was quite variable in the enemy mice at 3 weeks, as you can see by this very large standard deviation. And these differences were often less robust than those in the lymph nodes. Within the CD8 memory T cells, I noticed that the SPF mice actually had more memory T cells than the enemy mice at 2 weeks of age in both counts and frequencies. However, by 3 weeks of age, the enemy mice had surpassed the number of memory cells, had surpassed the number of SPF memory cells, and it was often at a pretty significant higher frequency. Within the lymph nodes, the number of memory T cells was consistently higher for enemy mice, both at 2 weeks and 3 weeks. And this difference only widened with age, as you can see, very significant differences by 3 weeks of age. And then in CD8 memory T cells, this difference was even more notable than within the CD4 memory T cells. As you can see, all the significance levels were below 0.001. And while there was a bit more of a similarity at 2 weeks of age between the SPF and enemy mice, this difference was quite robust by 3 weeks of age. Within the inflammatory monocytes, interestingly, the number of enemy cells actually decreased from 2 to 3 weeks in both count and frequency. And in the lymph nodes, we see a consistent increase and very significant differences between the number of SPF and enemy mice, with a lot more enemy counts and frequencies. So what does this all actually mean? The results revealed an increase in the number of cells in the memory compartment of the enemy mice compared to the SPF mice. And while the differences were often modest at 2 weeks, by 3 weeks there was generally an increase or significantly more memory cells in the lymph nodes in enemy mice compared to SPF mice. This means that mice with more microbial experience have a more developed immune response, or have a more developed memory cell response. And these findings were consistent with the hypothesis and also just with more basic biology. Since normal microbial exposure means that the mice are going to be exposed to a variety of pathogens, the mice need to combat these pathogens. And so it's logical that enemy mice would be developing or generating more memory cells than SPF mice. The results also revealed an increase in the number of inflammatory monocytes in enemy mice, which is associated with an increase in the production of inflammatory cytokines. This creates a more inflammatory host environment and is consistent with the findings in adult mice from Huggins et al. So essentially, this research demonstrates that infant mice are capable of generating an immune response similar to that of adult mice, simply from microbial exposure. And this implies that transmissible pathogens, rather than age alone, can influence the maturity of the immune system, since both the increase in number of cells in the memory compartment and the increased inflammation indicate maturation of the infant immune system. These differences call to attention the significant microbial exposure, the significant impact microbial exposure has on the composition of these cell profiles. So this project has revealed that microbial exposure significantly impacts the infant immune system within the memory compartment and the inflammatory monocytes. And in today's society, infants are exposed to a variety of pathogens. While the pathogen exposure may not be as diverse as enemy mice, they are not as limited as those of SPF mice, which draws to attention the need to develop better mouse models that represent the infant immune system. Through my reading, I came across Tao and Rees' proposed mouse model. which involves a primary screen with SPF mice, so similar to what current immunology research contains, and then a secondary screen using mice with pathogen exposure. If there are significant differences in the results between these two experiments, then it may be worthwhile to investigate the differences before continuing research in a human setting. And I think this method would be very effective in saving resources like time and money, since researchers are able to understand that if there are significant differences between these SPF and NME mice, chances are there will be alterations when converting to humans. And finally, the limitations of this project. One drawback to this project was the small sample size. 5 or 6 mice were used in each sample, and so a larger sample size would definitely reduce the chances of random error. Also, this study compared only 2-week and 3-week-old mice. I think comparing it to an adult sort of as a control or just something to serve as a baseline would be extremely helpful in understanding the relative maturity of these infant mice to that of an adult. So in my research, I found that microbial exposure leads to an increase in the number of memory cells and inflammatory monocytes in infant mice. And the mice compared were genetically identical. So that suggests that the environment and pathogen exposure are influencing the maturity of the infant immune system. And so, yeah, I'd like to thank Dr. Schultz for guiding me through this research process. His support and assistance was extremely helpful in understanding the methods as well as collecting data. And finally, these are my references. Thank you.
So Shreya, there are 3 questions that I'll be asking you for the oral defense part of your presentation today. And the first one is this: think back to the initial curiosity that sparked your inquiry. What other curiosities do you have, and how has this process prepared you to explore them?
So this process has definitely increased my curiosity into the infant immune system, and I think understanding more about it. Because as I went through this process, I realized that there were very few resources on the infant immune system, simply because of a lack of information about it. And that stems, I think, from weaker mouse models that make it difficult to understand infants, simply because there are ethical implications of analyzing an actual infant. So I think that this project has definitely increased my interest in understanding more about the infant immune system and gaining more knowledge about that.
And then can you tell me, how did you determine which results generated by your research method were the most important in forming your new understanding?
So as I said, there initially was a lot of data to look into and analyze. And so I ultimately decided upon the memory compartment and the inflammatory monocytes, simply because those were the ones I was most comfortable with. A lot of my preliminary readings and research had involved the memory compartment, like the CD8 memory T cells, were compared in one study to humans and mice. And so I think just simply my comfort with those topics translated very well into the infant, understanding that in the infant scale.
And finally, you did touch on this, but I was wondering if you could expand on it a little bit. How do your findings provide direction for future research in the field?
Yeah. So since current research only generally utilizes simply laboratory mice, especially in these infant models, I would propose doing 2 screens whenever research is conducted. So the initial screen would be what current immunology research uses, so the SPF or laboratory mouse model, but then maybe conduct that again. So if you're, so do the same exact experiment, but with NME mice, because oftentimes, especially with infants, when people are translating from the research to the clinical setting, many surprises or changes can happen, or unexpected things can occur, simply because the genetic the genetic information is very different from mice and humans, and then also the microbial exposure is very different. So I would recommend that second screening with NMEs.
Great. Well, this is the end of the formal part of your presentation with oral defense.